I'm Benjamin Blasco, the Senior Discovery Manager at GARPD, the Global Antibiotic Research and Development Partnership. GARPD is a not-for-profit drug developer focused on accelerating the development and access to treatment for drug-resistant bacterial infection. In our discovery and exploratory research program, we aim to discover and deliver novel preclinical candidates to address the urgent need for solutions against antibacterial resistance. Our model is to do that in collaboration with academic institutions and industry partners worldwide. Our focus is on bacterial species in the WHO critical priority lists such as Klebsiella pneumoniae and Acinetobacter baumannii. We prioritize new chemical entities that do not show cross-resistance to existing antibacterial classes and act through new targets or new mode of actions. We have developed a diverse portfolio of projects spanning 5 research areas, which are shown in the colored boxes at the top part of the poster. So in the small molecule area, we focus on identifying new hit compounds from diverse and previously unscreened libraries. Next, we're delving into untapped microbial sources to identify novel natural products with antibacterial activity. We're also searching for potentiators of aminoglycoside activity and efflux inhibitors. In the unrealized targets area, we are doing all the steps of target assessment and target-focused discovery. And lastly, we're also reevaluating undeveloped compounds whose development was stopped for non-scientific reasons. So earlier this year, we've published proof of principle data supporting our high-throughput screening strategy. And this study started in 2019 when we established the AMR Screening Consortium, comprised of 3 Japanese pharmaceutical companies, Takeda, Eisai, and Daiichi Sankyo. And based on the strategy developed and funded by GARPD, 3 compound libraries were selected from these companies and then screened by our partner at the Institut Pasteur Korea. So the left-hand side panel of the poster shows that compounds from the Eisai and Takeda libraries were selected following the entry rules criteria, which are associated with increased likelihoods of intracellular accumulation in gram-negative bacteria such as E. coli. So key characteristics of these compounds include the presence of ionizable amines, low globularity, and having 5 or fewer rotatable bonds. In the middle panel illustrates our generic high-throughput screening cascades that we used in this study and we are still using today. So starting from a little more than 48,000 compounds at start, 72 compounds showed confirmed activity against at least one of our primary screening strains, which includes 2 Klebsiella strains, 1 multidrug resistant, 1 multidrug sensitive, and 1 extensively drug-resistant Acinetobacter strain. So after removal of known chemical classes and unfavorable chemotype, 17 compounds were selected. These were purchased, quality checked, and then rescreened against our primary screening strains to confirm activity. And these were also counter-screened for cytotoxicity and hemolytic activity. Ultimately, 2 compounds from the Eisai library with activity against Acinetobacter specifically remained. And these are shown in the bottom right panel of the poster. And these hits, along with a selection of close analogs that were commercially available, were tested against a larger panel of Acinetobacter isolates and their kinetic solubility and microsomal stability measured. However, after a final medicinal chemistry review of the data package, the 2 chemical series were not pursued due to the lack of scope for optimization. But the key message of this work is the proof of principle that hits with activity against drug-resistant Klebsiella and Acinetobacter can be identified using this strategy. And indeed, since completing this work, we've screened over 150,000 additional compounds and nearly 4,000 microbial extracts using this strategy. And our discovery and exploratory research team is currently managing over 20 identification and E2Lead projects. Thank you.